Hello, I'm Maya Burton, and my final presentation concerns brain cell death in rats of different age groups after an incident of trauma. So, my research question is when both young rats of about 2 months of age and older rats that are fully neurologically matured both undergo an incident of brain trauma, after 1 month, which will encounter less neurological damage, measured in brain cell death. So, we can connect both our animal behavior and neuroanatomy modules to the foundation of this experiment. The animal behavior module, where we investigated the behavior of fruit flies after experiencing the treatment variable of ethanol exposure, is similar in that we're investigating the behavior of rats after trauma and documenting progress after a scheduled amount of time. Our neuroanatomy module is also helpful in that it provided methods in which we can analyze the brain and compare and contrast its parts. So I want to use that foundation to look into my research question. I first wanted to look at what occurs in the brain of a rodent when there is any sort of trauma to the brain, and what primarily happens is called apoptosis or cell death. However, I had to be careful in deciding what age group I would look at because there is an age range where rats naturally go through cell death in the brain just because that's how the brain develops. In that age group, that age range is up to about 10 days postnatally. So, as long as I was looking at rats past 10 days of age, that shouldn't have been a variable that interfered. And lastly, I had to decide how long after the incident of brain trauma I would look for changes in the brain. In a similar brain study conducted with 3-month-old rats, brain cell loss was looked at 1 hour, 2 hours, 48 hours, 1 week, 2 weeks, and multiple months, and then a year after traumatic brain injury. And damage was seen over a year later. So while using both old and young rats, I thought that 1 month was an appropriate amount of time to gauge which age group suffered the most cell death in the brain based on Smith et al. So, going into my experimental design, my official hypothesis is if a rat of about 2 months of age and a neurologically matured rat of about a year old both undergo traumatic brain injury, the younger rat will show more brain damage after a month than the mature rat. The treatment variable is their age. It's the only thing that differs between the two rat groups. The matured rat will be used as the control. The constant include the tunnel method being used to observe the cell apoptosis, the anesthesia they're put under, the method of traumatic brain injury, and the duration of time they're looked at after the injury. And this will be a replicable study. So my procedure is based on the same foundation as the study done by Rink et al., done on the 3-month-old rats. First, to standardize the traumatic brain injury, both rat groups will undergo anesthesia injections of sodium pentobarbital and hooked up to fluid percussion devices, which are devices that actually produce the brain trauma by injecting saline rapidly into the brain. This method of TBI is very similar to a human injury to the head. We take 6 micrometer pieces of coronal slices from the mid hippocampus region. And because I'm assessing damage 1 month after the injury, these slices are taken a month after the simulated head injury. I store them using paraffin blocks, which are just boxes you can use to store things for a large amount of times. This is the easiest way to assess the neurons in the brain. Seems to be with tunnel histochemistry, which is where terminal deoxynucleotide transferase deoxyuridine triphosphate, or DUTP, nicotine labeling, abbreviated as tunnel, detects apoptotic DNA fragmentation in the brain. The staining would be completed. We deparaffinize the samples and rehydrate them for viewing, kind of similar to what we did with myelin and Nissl staining. And then we can look at the brain using electron microscopy. In my results, I predict that we'd be able to see more cell death in various regions of the brain in the uh, mature rat. The areas of the brain I would expect to see more cell death is in the cortex, the hippocampus, a more concentrated of the area of the hippocampus called the dentate gyrus, and in the white matter of the coronal brain sections. This first visual is over to the right is a model of where we'd be looking at apoptosis in the brain. In this second visual is a more detailed version of where we'd be looking and where we'd find neuronal cell death. So cortex, hippocampus, white matter, and dentate gyrus. Okay, in the results, we see the areas where apoptosis would occur for both the young and old rat, but the reason why young rat brains would encounter more substantial consequences in the brain is because the brain is still developing to an extent. Developing neurons encounter this damage, and although the brain has great plasticity, this trauma is part of the brain as it develops. This hypothesis is supported by studies like the one conducted by Deng Bryant et al., where ketogenic diets prevented alterations in young rat brains after trauma, but not in old rat brains. And this leads me to believe that young rat brains are more susceptible to environmental change than adult rats. So there are a few reasons why studies like these are helpful in the grand scheme of things, including providing research on the human brain's ability to bounce back after physical trauma, especially in sports. Neuroscientists would use this study to answer questions about the long term effects of brain damage, how brain damage affects different age groups, and what happens after incidents of brain trauma. And here's a small look at my references.